I'm Raamesh, and thank you all for coming to my presentation. Today I'll be discussing my recent paper in PLOS computational biology. If you want to pull this paper up now or sometime later, you can search for active subnetworks discovery, and the top hit would be my paper. So let's get to the motivation for the approach we have developed. A canonical problem in genomics is that we have case versus control study. Like in microarrays or GWAS study, take several other types of studies. You have case and control, and you want to find out, you want to make discovery of that data. And this is an age where massive amounts of genomic data is being generated, and we have genomic data for the same experiment in other species. Also, we have networks for several other species, and we would like to make use of all this genomic data. What we need now are approaches, tools, and techniques and algorithms to integrate all this genomic data and to make sense of all of your experimental data. Let us consider one specific problem. We were interested in finding out what defines a stem cell, what makes a stem cell. So we had expression data of stem cells with respect to differentiating cells for both mouse and human organisms, and we wanted to discover what are conserved stem cell signatures in the two expression data. We started with a naive approach of comparing the top differentially expressed genes in both organisms, and we found like something around 15% overlap. So the overlapping genes are interesting, but we were looking at pathways or modules which are active in both the species. We were looking at a much higher level network structure. Also, we were looking at genes which were not necessarily differentially expressed, but they connected important genes for stem cells. So we decided to use networks in addition to the expression data, and the problem is we want to discover subnetworks of these networks, coherent dense subnetworks that contain genes which are active in both the expression data. So in this figure, green genes means these are genes active in stem cells, red genes means they are active in differentiating cells, and we have developed an approach to discover subnetworks of the functional linkage of the networks that contain active genes from the expression study. This area of integrating expression data with networks is not new. Ideker et al. As early as 2002 dealt with this problem. After that, there have been several studies which developed improvements for this algorithm, and they integrated protein interaction network with expression data for the single species context. In our study, the novel contribution is we are doing a cross-species approach. That is, if you have data in one species and if you can find data in other species, we have developed an algorithm to integrate these two expression data and use networks to do this integration. Also, with the massive amounts of data available in species, we can have much more comprehensive networks like the functional linkage networks, which these networks literally contain millions of edges and are of 2 orders of magnitude larger than protein interaction networks. We need new generation of tools, and we have made sure that our algorithm is scalable to the functional linkage networks. I will describe my approach. We call our approach Nexus, and we start with the functional linkage networks for both mouse and human, and we start with genes that are differentially regulated in both species. We start with each gene in this set, and for each seed gene we find its functional neighborhood. Functional neighborhood contain all those genes that are within a certain product distance from the seed gene. So in this case, the seed gene is 1, and the gene 12 is product distance 0.8 multiplied by 0.8, that is 0.64, which is greater than certain threshold. That's why it's in the functional neighborhood of that gene. Likewise, in both the species, all the genes are in a certain product distance. Now, once we have the functional neighborhood, we will overlay the fold change from the expression data. And again, green means active in stem cells, red means active in differentiating cells. And we want to find out coherent subnetworks that are densely connected and at the same time contain lot of active genes from the expression data. So we start with a seed gene 1, and we will incrementally add genes in a greedy fashion so as to maximize the average activity score of the subnetwork. For this cross-species setting, what we do is we calculate a gene score for each gene. We will so if the two genes show similar behavior, they will have a large score. If they show opposite behavior, they will have a small score, and we would not like to add them in the subnetwork. And we average the gene score for all the genes, and that is what I am calling the average activity score. At the same time, we want to make sure that these subnetworks are densely connected. So I put a constraint on the clustering coefficient of the subnetwork, and that ensures that there are a lot of that the subnetwork we discover is cliquish. So we incrementally keep on adding genes to the subnetworks, and we continue this till the clustering coefficient of either of the subnetwork does not satisfy the constraint, or the average activity score of the subnetwork has gone below certain value. When we applied our algorithm Nexus on the stem cell data, we discovered more than 200 distinct subnetworks. Average size of these subnetworks was 21, and most of the subnetworks were enriched for at least one function. We further looked at the subnetworks which contain lot of genes which are active in stem cells, and we found them to be enriched for functions like cell cycle, DNA repair, gene transcription, and regulation. In contrast, we also looked at subnetworks that were enriched for differentiating genes which were active in differentiating cells, and they were enriched for growth, development, and differentiation. This is what we expected, and we saw that. This is one example subnetwork which we discovered. This subnetwork is interesting for in cell cycle context. CCNB1, MYBL2 are important cell cycle regulators, and CCNF is also known to play a role in CCNB1 localization in nucleus. So in general, we discover subnetworks where we know that there are few genes which are interesting for whatever context you are interested in, and there are other genes which we don't know about, and we can develop hypotheses to do experiments on. That is the power of this approach. Now to validate that the subnetworks we are discovering are significant, also to compare that our method with other methods, we we start with a single species case. I'm making a switch from the cross species case to the single species case so that I can compare with other methods. So here I am showing as the average activity score progresses, we are discovering less and less number of subnetworks. That is what we expect. As we increase the stringency, we discover lesser subnetworks. But we wanted to also make sure that the subnetworks we have discovered are significant. They are not by chance. So what we did was we used the differential gene scores for the actual real case, and we randomly placed on other genes on the network. And our question is that for the real case we should discover lot many subnetworks than for the random case. We do this randomization like 5 times, and I plot them here. Here we see that for the real case we discover lot many subnetworks than for the random cases, and that is good. But one thing that would be surprising to you is even for the random case we can discover lot many subnetworks, and this is not limited to my approach, but this is a problem faced by other approaches as well. But our approach we can do quite reasonable in finding real subnetworks. Now, when I compare our single species approach with the cross species approach, the first thing you will notice is that the difference between the real and random subnetworks is much more in the cross species case. In the real case, it's like 2-fold, 3-fold. In the single species case, it's like 2 or 3-fold, and for the cross species case, it's like around 20-fold. So, doing cross species analysis, using your data with other species is good. Why is this happening is we are using coherency constraint that a particular subnetwork should be conserved in the both species as well as should contain active genes from both species. So this coherency constraint limits the number of random subnetworks, and we are able to get lot many significant real subnetworks. Also, if I give you a particular subnetwork which shows similar behavior in two species, then it's a lot more interesting to you to look at and to develop hypothesis for. So I have discussed I have discussed my cross species problem where we integrated functional linkage networks with expression data. I would like to tell you that this is not limited to expression data or functional linkage networks. We can use other networks and other data, like we can use copy number variation or GWAS scores. It should be so. We can do it for other kinds of genomic data. Also, we modified our approach for the single species case so as to compare with other algorithms. So if you have data in only one species and have one network, we can do it. Also, another interesting thing is we can find 10 subnetworks in the two species, but these subnetworks shows different behavior in the two organisms. That is also possible with Nexus, and I have showed you one example case where if you look at the genes, they show opposing behavior in the two species. Also, other cases are also possible when you have two networks and one data set you want to use network of another species. We can do it. If you don't have network in your species, then we can integrate with network of another species and data set of another species. That is also possible. So I'd like to thank my advisor Chad Myers. I also would like to thank my collaborators Shika, Veshuhu, and Catherine. I like to thank the ISMB Travel Fellowship and MPGI Travel Fellowship for supporting my travel here. And I take this time to also advertise for postdoc positions in our lab. If you are interested, you can talk to my advisor Chad Myers. He's at the ISMB, and yeah, you can talk with him. And if you want to take a look at the figures, and if you or my algorithm source code, you can search for active subnetworks discovery, and the top is my paper. You can go to supplementary, and you can find all the code required for it. If you want any help, then you can contact me or Chad, and we will be glad to help you. So thank you all for listening to my presentation, and at this point I would like to take questions.